So right now we have these artificial intelligence models that can basically dream up a theoretical cure for almost any disease. But there's a massive catch.
Right.
The tools just, they don't actually talk to each other. We're at this point where computational biologists are spending way more time just writing custom Python scripts to move data around between different software environments than they are, actually engineering proteins. It's just a massive bottleneck in drug discovery right now.
Yeah, and it is a profound structural problem in the field. When you look at the current stack of structural biology software, you realize pretty quickly that the fragmentation isn't just like an inconvenience. It actively breaks the foundational sequence-structure-function relationship.
Which is?
We're talking about Anfinsen's dogma, right?
Yeah, exactly. Anfinsen's dogma.
Right, the principle from the 1960s. The idea that a protein's linear amino acid sequence completely dictates its 3D folding, which in turn dictates its biological function.
And for you listening, think of it kind of like a string of letters that magically folds itself into a very specific tool, like a tiny pair of scissors or a lock.
Yeah.
But right now, that triad sequence, structure, and function is scattered across entirely different servers.
Exactly.
You've got AlphaFold taking a sequence and generating a structure. Then you have to export that PDB file, write a script to reformat it, and then feed it into protein MPNN to go from structure back to sequence.
Yes.
Or, you use RF diffusion to go from function to structure. None of these paradigms share a native environment. It's honestly like having a state-of-the-art kitchen, but every single gadget speaks a different language, requires a totally different electrical plug, and there's literally no counter space to actually combine the ingredients.
That is a perfect analogy. And that brings us to the core of the research we are diving into today. A team from Genentech Research and Early Development has published a really fascinating paper introducing GYDE. That stands for Guide Your Design and Engineering.
GYDE.
Yeah, and it's an open source, web-based platform designed specifically to unify that totally fragmented workflow. The goal is to create this seamless environment where the AI computations, the structural visualizations, and the experimental data all just live in the exact same contextual space.
Okay, so the architecture behind how they actually pull this off is what really caught my attention. The magic word throughout this entire paper is no code.
Oh, yeah, huge selling point.
Right. You access GYDE through a standard web browser, but under the hood, it's built on a modular architecture using a centralized columnar data frame that's backed by MongoDB.
And that database structure is critical here because biological data, it isn't just text. You're dealing with these incredibly complex matrices of atomic coordinates, confidence arrays, and evolutionary sequence alignments.
Right. It's massive.
It's huge. So by using a MongoDB-backed columnar format, GYDE can pull massive data sets from public repositories like the Protein Data Bank and UniProt and just instantly sync them alongside internal lab data without any formatting conflicts.
And the way that data is presented completely changes the analytical workflow. Like you have a multiple sequence alignment viewer, an MSA, sitting right there directly adjacent to a mole structural viewer.
Yes.
So if you highlight a specific residue variation in the text-based alignment, it instantly illuminates that exact coordinate on the 3D model. The visual interface physically restores that sequence structure connection that the disparate AI tools had basically broken.
It's beautiful to see. But, the visual integration raises a really massive technical hurdle.
Yeah, for sure. The compute power.
Exactly. The deep learning models required for these predictions, the AlphaFolds and the MPNNs, they rely on massive clusters of GPUs, graphical processing units. A standard web browser simply cannot handle that kind of computational load.
I mean, if you try to run an AlphaFold multi-time prediction locally on just a standard machine, you're going to be waiting days, right? Assuming the system doesn't just completely crash from memory exhaustion.
It definitely would crash. And that is where GYDE's integration of the Slivka Compute API becomes basically the linchpin of the whole platform.
Slivka. Okay, explain how that works for us.
So Slivka acts as an asynchronous job scheduler. When a researcher configures an experiment in the GYDE browser interface and hits run, Slivka packages that request, offloads it to the high-performance computing clusters, manages the entire queue, and then just streams the heavy output back to the web dashboard.
Wow.
So the user's actual machine does virtually zero heavy lifting.
It is entirely decentralized computation mapped to a centralized visual hub.
That's brilliant.
Yeah.
And because the entire workspace state is managed in that database, you can just generate a URL and send your exact visual layout structures, plots, sequence alignments directly to a colleague.
Yeah, they open the link and see the exact same 3D coordinates and data frames that you are looking at. Okay, so let's do a quick recap for you listening, just to make sure we've got the foundation. AI tools are amazing, but they're terribly fragmented. Genentech built GYDE, a no-code dashboard in your browser to fix this. It uses MongoDB to manage the data, and Slivka to offload all the heavy math to supercomputers. So basically, anyone can run world-class AI biology without writing a single line of code.
That sums it up perfectly.
Which brings up a great question to keep in the back of your mind as we go, what happens to drug discovery when anyone, anywhere, can run these tools? But okay, let's move from the architecture to what this infrastructure actually allows scientists to uncover in the real world.
Right. Let's get into the benchmarking.
Yes. The Genentech team benchmarked GYDE against a series of massive structural biology challenges, starting with the interactome, predicting how nature works at an incredible scale.
They specifically looked at single-pass transmembrane proteins, or STMs. These are these critical cell surface proteins that manage communication, and they often function by forming multimers, which are just complexes with other proteins.
And the scale of the experimental proteomics data they fed into GYDE proposed, like 1,381 putative interactions between these STM proteins.
Which is a staggering number. And to give you some context on the knowledge gap here, only about 3% of those complexes had ever been physically mapped with experimental structure.
Just 3%.
Exactly. So they deployed AlphaFold 2 multi-inter across all 1,381 potential complexes to see if the AI could predict the physical docking interfaces.
And this is where GYDE's native plotting tools turned raw data into just an immediate biological insight. They generated 5 separate structural models for every single interaction, right?
Yes. And then they utilized GYDE to plot a very specific metric. They graphed the maximum confidence score of the AI against the standard deviation of the confidence scores across those 5 models.
And the mathematical logic there is just brilliant. For you listening, think about it like this. If the AI is highly confident, but the 5 models vary wildly from each other, meaning a high standard deviation, the folding trajectory is likely unstable or artificially forced.
Right. It's just guessing confidently, but inconsistently.
Exactly. You are looking for a high signal and low variance.
Yeah.
So when they plotted this in GYDE, what did they see?
The resulting scatter plot instantly revealed a distinct cluster. The Genentech team actually termed it the blue box region.
The blue box.
Yeah. These were novel protein interactions with remarkably high confidence scores and incredibly low standard deviation, meaning the AI was arriving at the exact same highly confident structural docking interface every single time it ran the simulation.
So GYDE basically filtered out the noise of over 1,000 unverified interactions and just handed the lab scientists a highly concentrated list of novel complexes with the highest probability of physical validation.
Precisely. It turns weeks of manual data sifting into a single glance at a scatter plot.
But that same visualization engine also exposes the dark side of these models, particularly when we start talking about AI hallucinations, which is a huge issue.
It is a critical vulnerability in computational biology right now. The Genentech team used GYDE to run a reality check on 2 newer AI co-folding models, Chai-1 and Boltz-1.
Right. They
used a benchmark dataset called runs and poses,
which is specifically designed to test the boundary of the AI's training manifold, right?
Yes, exactly. The dataset challenges the models with targets that are increasingly dissimilar to anything the AI actually encountered during its training phase.
Kind of like giving a student a test on calculus when they only studied algebra.
That's it. And when they plotted the predictions inside GYDE, the drop-off in accuracy was just starkly visible.
The scatter plots prove that when Chai or Boltz are asked to predict a structure that shares evolutionary homology with their training data, their accuracy is phenomenal.
It's great.
But the moment you push them into completely unseen structural territory, they completely fail to generalize. They aren't really learning the underlying physics of protein folding. In many cases, they are simply memorizing and interpolating from the protein data bank.
And having a platform like GYDE make those blind spots highly visible is honestly just as valuable as finding the successful predictions. It prevents researchers from spending months synthesizing and testing a protein complex in the lab that the AI simply hallucinated.
Okay, so another quick summary here. Case study 1. They used a blue box plot to find reliable new protein connections out of over 1,000 guesses. Case study 2.
Yeah.
They used GYDE to catch AI models cheating, basically proving they memorized data rather than learning true physics.
Right.
So for you listening, the question is, how much should we trust an AI that hallucinated as cure? But let's keep going, because predicting how nature works is one thing, but the real power of a unified platform unlocks when things go wrong in the lab, when you have to modify or fix a structure. Let's look at how GYDE handles antibody engineering.
Yeah, this is where it gets really practical. The paper details the optimization of an anti-SARS-CoV-2 antibody known as CO240. The goal was to enhance its binding affinity to the viral spike protein. So they started by pulling massive amounts of human B-cell repertoire data from the OAS database directly into the GYDE environment.
So they were looking at how the human immune system naturally mutated homologous antibodies during active COVID infections out in the real world.
Exactly. And GYDE renders this data as this beautiful interactive heat map, mapping the frequency of specific amino acid substitutions across the antibody's complementarity determining regions.
The CDR loops.
Right, the CDR loops that actually physical grip the target.
And a glaring pattern emerged on the heat map at position 98 on the heavy chain CDR3 loop.
It was undeniable.
The human repertoire data showed almost zero tolerance for positively charged amino acids at that specific coordinate. Conversely, it showed a really high frequency of negatively charged residues, specifically aspartic acid and glutamic acid.
And you can instantly deduce the electrostatic potential landscape just from looking at that heat map. If the antibody's binding loop actively selects against a positive charge over millions of evolutionary iterations, it strongly implies that the target epitope on the viral spike protein must possess a dense positive charge.
Because two positive charges would create severe electrostatic repulsion, like pushing two magnets together with the same pole. It would completely kill the binding affinity.
Exactly. So to confirm that deduction, they ran a bodybuilder 2 entirely within GYDE to generate the 3D structure of the C1Gel240 antibody.
Oh, wow.
And the mole viewer confirmed that position 98 was highly solvent exposed, meaning it was facing perfectly outward to interface with the antigen. Cross-referencing this with the viral structure revealed lysine 417, which is a positively charged residue on the spike protein, sitting directly across from position 98.
That is incredible. The evolutionary data perfectly predicted the physical electrostatic interaction. And because of the unified interface, the researchers didn't even have to switch to another program to act on this, right?
Nope. GYDE features a built-in picklist generator. They could select the optimal negatively charged mutations directly from the heat map and just export the sequences for physical synthesis right there.
So the friction is totally gone. That demonstrates optimization, but the platform's utility is arguably even more critical when rescuing a failed asset. Let's talk about the case of the H1340.cc rabbit antibody.
Oh, this is a classic developability nightmare. So this molecule was designed to target PD-1, which is a major cancer immunotherapy receptor.
And the binding affinity was great.
It was fantastic. But it had a severe liability for large-scale manufacturing. It contained a non-canonical disulfide bond.
Okay, let's break that down for the listener. Disulfide bonds are crucial for structural rigidity, but a non-canonical configuration means the cysteines are positioned in a way that is atypical and often thermodynamically unstable during the really complex scaling process of drug manufacturing.
Right, you might be able to make a tiny bit in the lab, but you can't brew vats of it for a clinical trial.
Exactly. So the obvious laboratory solution is to simply mutate out the cysteines to remove the problematic bond.
Which is what they tried. But when the lab scientists did that, the antibody completely lost its ability to bind to PD-1.
Oh, wow. Why?
Well, the bond was apparently imposing a necessary steric constraint on the CDR loops. Removing it caused an entropic penalty. The loops basically became too flexible, like a wet noodle, to actually lock on to the cancer target.
They were trapped in a classic optimization paradox.
Yeah.
Fix the manufacturing, kill the drug. Fix the drug, you can't manufacture it.
Exactly. To resolve it, they uploaded the antibody's coordinates into GYDE and initialized protein MPNN. The objective was to redesign the sequence around that specific region to maintain the rigid backbone conformation without actually relying on the non-canonical disulfide staple.
And protein MPNN generated a huge array of potential sequence solutions. GYDE's frequency analysis widget then processed those outputs and revealed the statistical distribution of the AI's logic.
Right. And the highest frequency suggestion, which appeared in about 22% of the generated proposals, was to mutate the cysteines to an ala-ala configuration. 2 alanines.
And did it work?
When they synthesized the ALA-ALA variant in the physical lab, the results were completely definitive. The mutation successfully stabilized the local backbone structure, completely restoring the high binding affinity to PD-1 while permanently eliminating the manufacturing liability of the non-canonical bond.
That is wild.
Okay.
Right, time for a quick midpoint recap. Case 3 showed us how GYDE uses evolutionary heat maps to deduce the shape and charge of a virus, basically finding the perfect puzzle piece to neutralize it.
Right.
Case 4 showed how they used AI within GYDE to surgically repair a broken rabbit antibody, swapping out a bad chemical bond with 2 alanines to save a cancer drug. So, listener, think about this.
Right.
If we can fix nature's mistakes this easily, what happens when we start building entirely new things from scratch?
Which brings us to the final frontier of this technology, de novo design, building entirely novel proteins from scratch that have absolutely no evolutionary precedent.
Yes. The paper demonstrates this capability by replicating the engineering of the hyper-TeV protease. The original biological objective here was to take the natural TeV protease, which is an enzyme that cleaves specific peptide sequences, and engineer a variant with substantially higher catalytic efficiency. They wanted to make it like 20 times better.
Right. So the Genentech team loaded the wild-type protease structure into GYDE. Using the visual interface, they carefully masked the active site.
Masking the active site, meaning the specific residues responsible for the actual catalytic cleavage. You don't want to mess those up.
Exactly. They protected the scissors, essentially. Then they instructed Protein MPNN to redesign the broader structural scaffolding while keeping that active site strictly untouched.
And the parameter tuning here is what's truly fascinating to me. They set the AI's sampling temperature to 0.2 and introduced a degree of backbone noise. Let's explain that temperature parameter because it's so important for these models.
Yeah, go for it.
So in the context of these machine learning models, the temperature parameter controls the softmax distribution of the output probabilities. A higher temperature flattens the distribution, leading to wild, highly divergent, almost jazz-like sequence generation.
Right, it gets very creative, but risky.
Exactly. But dropping the temperature to 0.2 sharpens the probability mass. It forces the AI to make highly conservative, thermodynamically stable predictions for the scaffolding, like playing sheet music exactly as written.
That's a great way to put it. The combination of that low temperature and the backbone noise allowed the model to explore stable alternative packings without disrupting the global fold.
And the results?
Well, when they compared the variants generated by GYDE in literally a matter of minutes to the physical hyper-TeV variants that scientists had previously spent months developing through painstaking lab work,
over 95% of the AI's predicted mutations perfectly matched the successful real-world designs. It effectively automated months of directed evolution with a few clicks.
It's staggering.
But my absolute favorite application in this entire paper deals with the messy, multivariable trade-offs of in vivo biology. They used GYDE to engineer a targeted gene therapy delivery vehicle.
Yes. The LRRC15 project. The objective was to design a de novo mini-protein that binds with really high affinity to LRRC15, which is a known cancer-associated fibroblast biomarker.
Basically a molecular flag that says cancer is here.
Exactly. But a binder alone isn't a therapy. They needed to fuse this mini-protein to the capsid of an adeno-associated virus, or AAV, which acts as the actual payload delivery vector.
Which is an incredibly complex engineering challenge because you are modifying the exterior of a viral shell. The Genentech team generated thousands of candidate binders using 2 distinct generative models, RF diffusion, which builds structures from spatial constraints, and Bindcraft, which approaches the problem through sequence and motif optimization.
They narrowed that massive pool down and physically evaluated 175 of these designs in the lab. They measured 2 critical independent variables. First, the binding affinity of the mini-protein to the cancer target, and second, the actual viral yield.
Meaning how well the AAV managed to assemble its viral shell with this new clunky mini-protein grafted onto it.
Right, so they took all that physical assay data and fed it back into the MongoDB data frame inside GYDE.
And when they plotted the 2 variables against each other, the dashboard immediately visualized a brutal biological compromise.
Yeah, the scatter plot revealed a really stark divergence based on the generative model they used. The mini-proteins designed by Bindcraft exhibited exceptional binding affinity to the cancer biomarker.
They gripped it tight.
They did. However, their structural properties severely disrupted the AAV capsid formation. It resulted in just abysmal viral yields.
It's like designing a sports car that goes 200 miles an hour. but it has literally no trunk space to deliver the cargo. And the RF diffusion designs were the exact inverse, right?
Yep. They integrated beautifully with the viral shell, keeping manufacturing yields incredibly high, but they lacked the necessary affinity to effectively grip the cancer target, like a minivan with a weak engine.
So in a standard workflow, navigating this kind of multi-objective Pareto optimization problem across hundreds of candidates requires extensive data manipulation. It's a nightmare.
Oh, it takes forever.
But GYDE's plotting interface allowed the team to visually map that Pareto frontier instantly. They could just scan the plot for the rare candidates that successfully balance both high viral yield and sufficient binding affinity, the Goldilocks zone.
That precise structural compromise is exactly what is required for a viable clinical asset.
It is the perfect demonstration of why computational tools need to be unified. The AI didn't solve the LRRC15 problem entirely on its own. It offered two extremes. It required the human scientist to evaluate the biological trade-offs. GYDE simply removed the friction of analyzing that data so the scientist could actually make the decision.
Right. It empowers the human rather than replacing them.
Okay, final mini recap before we look at the future. De novo design, case 5. AI perfectly predicted in minutes what took scientists months to evolve for the hyper-TV enzyme, case 6. GYDE helped researchers find the perfect compromise between a cancer-hunting protein and the virus delivering it.
Excellent summary.
Which brings us to the ultimate question. Where does this go next? The paper outlines the integration of large language models, LLMs, directly into the GYDE architecture.
Yes, and that will fundamentally alter how scientists interface with these tools. We are moving rapidly toward prompt-based protein engineering.
Meaning you won't even need to manually select the masking residues or adjust the temperature parameters in a dialog box.
Exactly. A researcher could simply type, design a de novo binder for this LRRC15 epitope, optimize the scaffolding for AAV integration, and plot the predicted yield against affinity.
And the LLM will just parse the biological intent, translate it into the required API calls, manage the Slivka job execution across the entire HPC cluster, and render the final visualizations right there in the browser.
It is the ultimate democratization of sequence structure function engineering. The technical barrier to entry for world-class computational biology is just dissolving before our eyes.
You literally no longer need a deep background in command line execution or Python pipeline development to design functional therapeutic proteins.
Which I think leaves us with a necessary and really complex question regarding the future of this field. Platforms like GYDE are effectively removing all the friction from rational protein design. They are packaging the most advanced physics and machine learning engines in the world into a point-and-click browser interface.
So the bottleneck is shifting from computational expertise to simple biological imagination.
Exactly. But as that barrier to entry drops all the way to zero, we have to consider the broader implications.
Right.
If any researcher, anywhere, can seamlessly translate a desired biological function into a physical, synthesizable protein structure with just a simple text prompt.
What does that mean for the world?
Yeah. So I want you listening to think about this. If you had the power to seamlessly translate biological function into physical structure with a single click, what is the first disease you would try to engineer a cure for? How do we prepare for a world where the power to engineer biology is completely democratized?
That is an incredible thought to leave on. Thank you for joining us on this deep dive.